day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Vedika Maraj, and this lesson is brought to you by the Gauteng Department of Education in collaboration with Cybono Discovery Center. Today, we're going to be doing a lesson on scientific investigations. Now, scientific investigation seems to be a very daunting topic for life science learners, and in grade 12, we reached the point now where we are going to see it in both our final examination papers, paper 1 and paper 2. You will get a scientific investigation. Usually, the scientific investigation is about 10 to 15 marks. It will be one in paper 1 and one in paper 2. Sometimes it's broken up into two smaller investigations in the paper, but you definitely are going to get one in paper 1 and in paper 2. Now, the unfortunate thing about it is that you can't really study for a scientific investigation because it's not really content based. They can ask you a scientific investigation on any content. So this is where our learners struggle. But the good thing about it, the good news about it, is that it's a skill. And how to answer the questions on the investigations is a skill. You're going to be getting an investigation that you've never seen before. Okay? It's going to be one that you haven't seen before. But the main thing that you need to remember are the points that we are going to be discussing with you today. So when you get a question paper as a scientific investigation, this is what it's going to look like. This is one of the examples of what a scientific investigation looks like. Right? So they're going to give you a little stem, they're going to give you some information. They usually tell you how the investigation is conducted, and then they give you the results. The results can either be shown as a table or as a graph. Sometimes they don't give you the method. They just give you graphs with some information on the investigation. They do differ. The best way to combat this is, like I said, to understand the skills, understand how to answer the questions that they're going to give you, and also to practice. Practice, practice, practice. The more scientific investigations you do, the easier they will become for you to answer them. The more exposure you have to it, the better it is for you to deal with it. Right, so let's read through this investigation. So what you do is, first thing you do when you get an investigation is that you read through it carefully. You read through it with understanding. When you are done, you should be able to ask yourself, what is this investigation about? And you should be able to answer yourself. Then, when you're reading it again, you should now be thinking, or even the first time when you read it, you should be thinking about these different points, okay? I'm going to flag them as after we've read the investigation. An investigation was carried out to determine the influence of alcohol on the volume of urine produced. 12 healthy 23-year-old males of similar height and mass participated in the investigation. The investigation was conducted as follows. The men were divided into two groups of 6 each, Group A and Group B. The two groups ate the same food and did the same exercise over the 24 period before testing. Each group was given the following to drink after the 24 period. Group A was given 1 liter of alcoholic beer, which is beer that does not contain alcohol, and Group B was given 1 liter of alcoholic beer. And the urine was collected from each man every hour. Assume that the volume of urine collected is equal to the volume of urine produced. The results of the investigation are shown in the table below. So they're giving you some results here. Now, when you're reading it, these are the points that you need to think about. So firstly, you need to ask yourself, what is the aim of this investigation? You need to be able to identify the aim. The aim is what the investigation is about, or what they are investigating. Okay? And how do you determine the aim? You look for the words to determine or to conduct, right? That is how you identify. So in this instance, the aim is to determine the influence of alcohol on the volume of urine produced. From that aim, you will be able to identify you should be able to identify the independent and the dependent variable of the investigation. So most often, the first question they're going to ask you in a scientific investigation is to identify the independent and the dependent variable. And you get this from the aim. So you look at the aim, and that's going to give you the two variables. Now, the independent variable is the one that the investigator controls and can be changed. Whereas the dependent variable is the one that is changing due to or because of the independent variable. So it is dependent on the independent variable, right? So in this instance then, what would be our independent variable? It would be the alcohol, right? And our dependent variable will be the volume of urine produced, right? The next thing we need to ask ourselves is, how did the investigator ensure reliability? When we're talking about reliability, what are we talking about, right? Think about it. What are we talking about? And also, I want to bring to your attention, I want to put a big asterisk here, because if you really look at the last few years' papers, whether it's paper 1 or paper 2, this is one of the questions that are almost always asked. So, when it comes to reliability, we want to know what makes an investigation reliable. What's going to make our results reliable? So, let's think about it a little bit. If you increase your sample size, which means that you would have a larger sample or you have a large sample, that would increase the reliability because it would tell you that this is not just a fluke, okay? We didn't just do it with 2 participants and therefore it occurred because of any variable. We used a large sample, so our result is reliable. Another way is to repeat the investigation, because if you do the whole investigation again and you get the same result, or you do the same investigation 5 times and you get the results, then it is a reliable, the results are reliable. Why? Because it's not that you just did it one time and it could be a fluke or it could be because of any other variable. Then another one, it could be taking many measurements. So that it's not that I only took 1 or 2 measurements and now I'm coming up with a conclusion for this whole investigation. We took many, many measurements. They will tell you how many. But now, when it comes to questions on reliability, the examiners want you to answer and be specific. You cannot always just answer, increase the sample size, take many measurements, and repeat the investigation. You have to look at what they have given you in the scenario. So in this instance, let's look at the things that we had. Firstly, there were 12 healthy males that were used. Okay, 12 is a fairly large sample size, but if you're not convinced by that, let's carry on reading to see what else could be our reliability. What about this one here? The urine was collected from each man every hour. The fact that it was done every hour, and as you can see here, it was done, there was at different, it was taken for every hour for 3 hours, right? So that increases the reliability, right? Because it was done every hour. There's nothing here that they did that indicates to us that this investigation was done repeated. So if the examiner now has to ask you, how can they ensure reliability? Now you need to think to yourself, okay, there was a sample size that is fairly large. The urine was collected every hour, so that is the measurements that were taken many times. But they did not repeat the investigation. So then you can say, do the investigation more than once. Repeat the investigation and conduct it more than once. They want you to be specific. Then we come to validity. Validity is about fairness. It's different from reliability. Validity is about fairness. What makes the investigation fair? And it's that you are doing the same thing to both groups. So what did we apply here? That we applied to group A and we applied the same conditions to group B. Because you cannot, when you've got group A and group B, you cannot compare the results if you don't apply the same variables to both, constant variables to both. And you must be able to be fair. So this is when we are keeping the things the same. So what did they have here? We're talking about similar height, similar mass, similar height, similar mass, the fact that they were all male, so that's gender, the fact that they were the same age. So here we have same age, we have same gender, we have similar height, we have similar mass. Those are all examples of validity. The fact that they were divided into 2 groups of 6 each. Okay, so that is also validity because they put 6 in the one group and 6 in the other group. It wasn't uneven where they have 10 in the one group and 2 in the other group. That is all about fairness, which is validity. Then we've got the control versus the experiment. Right, so the control is the one, you have 2 setups. You get a control setup and an experiment setup. The control setup is the setup without the factor or without the variable that is being tested. The control is the setup without the variable or the factor which is being tested. So in this instance, the influence of the alcohol on the volume of urine is being produced. That is the influence of the alcohol. So the variable, the control is going to be the one that does not have alcohol. So group A in this instance is the control because it does not have the alcohol. And then the experiment is the other one. Why do we have a control? We have a control so that we have something to compare to. So that in the experiment, you can prove that this experiment, the result of this experiment is due to the fact of the alcohol. The alcohol is really the factor here that is affecting this, because in the control, we do not have that factor. So you can compare what happened from the control to the experiment group. Then you've got the control variables. This is basically similar to validity, where they ask you which variables were kept constant. In this instance, you don't always have to say same. You can just say age, gender, etc. When it comes to validity, asking you questions, how did they ensure validity? Then you will have to tell us from the extract, they use the same amount of number of people, they use the same gender, etc. Also with validity, they can ask you in the future tense, they can ask you how can they ensure validity, and then you mention things that are not in the investigation. Right, so I basically summed up everything for you over here. But now, with regards to, I'm going to go into a little bit more detail and touch on a few things that I didn't mention earlier. So there are three types of variables. Independent, one thing I forgot to mention. After you have now thought about all of these things, identified and highlighted, can you see what I did there? I identified and highlighted things. You need to start thinking about these terms, as I said. Now, when you get to the questions, it's going to be easier for you because you've already identified half of the things that they're probably going to ask you. Right, so the independent variable, like I said, is controlled or changed by the investigator to determine what effect it has. And in a graph, this is the x-axis. Whereas the dependent is the effect of the independent variable, the response that is measured or monitored during the investigation, and in a graph, it is the y-axis. But like I said, however, when you are asked for identifying the variables in a scientific investigation in your question paper, you cannot just look at the graph or the table that is given you, because that most of the time is just the measurement it is measured in. If you need to draw a graph from this table, then you will be able to use the results table to get your independent and your dependent variable for your graph. However, when it's talking about the independent and dependent variable of the investigation, you need to identify the aim, and that is where you'll get those variables. Then we come to a fixed or constant variable, all the factors that must be controlled or fixed when conducting an investigation. Okay, so we spoke about these. And then as this speech bubble I have over here is reminding you that you need to look at the aim of the investigation to identify the independent and dependent variable. Right, reliability. We spoke about this, but let's just look at what we said and see that we've covered everything. So as I said, the idea behind reliability is that any significant results of an investigation must be more than once-off finding. It must be repeatable. It cannot just be a once-off thing. Other researchers must be able to perform exactly the same experiment under the same condition and generate the same results. In questions which ask learners to state how the reliability of the investigation could have been improved, the correct answers could usually be the following: repeat the experiment, the investigation, or increase the sample size. But, like I said, be more specific. So if they said that only 1 or 2 people were used, then you should say, use more than 2 people so that you have a larger sample. Be specific. If they asked you, there were 200 pigs that were used, then you cannot just say a large sample size was used. You need to say 200 pigs were used, which is a large sample size. Okay, you need to be specific. Or take many readings and calculate the average, right? As I said, you need to say take many readings. Or if you saw that they only took 1 reading, then you say take more than 1 reading. Validity questions how the experiment or investigation was carried out. It's about fairness of the investigation if all the variables are controlled except for the variable being tested. And in these questions, some factors that might have decreased the validity of the investigation. So sometimes they can ask you to criticize the investigation. They can ask you, why is this investigation not valid? And they've been doing that recently. They've been asking learners, why is this investigation not reliable? Why is this investigation not valid? So now you need to actually criticize it and you need to look at the variables. For example, you would say that some factors that were some factors or variables were not fixed when carrying out the investigation. So they did not apply they did not have constant variables. Right. So I mentioned this as well. You need to look at the tense of the question. So the past tense would be, how did the investigator ensure reliability? That is the past tense. So what did they do? In that instance, you need to actually look at the answer. The answer will be provided in the question, right? So what was already done in the investigation? Whereas the future tense would be, how would they ensure reliability or validity? And that answer must be determined from the information. So you need to figure out what should have been done in the investigation. Now, grade 12. If the question is about pigs, you cannot be talking about humans. You need to be specific to the question. Okay, whatever you say in how they should ensure reliability and validity, it must be in context of the investigation that is given to you. Right. Now, when it comes to this one, right, these are the scientific processes, and this is frequently asked in life sciences, right? And you must be very, very careful when you are reading these. Careful and repetitive reading of the STEM and investigative process is required. And these are what the terms mean. So I've basically done most of them with you. So I'm not going to go through all of them, but I just want to indicate observation. An observation is what the scientists saw, heard, or encountered that encouraged them to investigate further. Okay, so it is an observation that was made, and from that observation, a hypothesis was made. And the hypothesis is a possible prediction or explanation and of the relationships between two variables. Okay, so observation is something that was seen or heard, and then they decide to investigate further and come up with a hypothesis. And then you have an aim. What did they investigate? You have the independent and dependent variables. Then you've got the controlled variables. The controlled variables we spoke about. We spoke about the control. Let's read quickly about the control. A second setup in the investigation that allows a comparison with the results of the experiment. Important word. The control is identical to the experiment, except that it excludes the variable that is being tested. What is accuracy? When we talk about accuracy, we refer to the care taken when taking the measurements, when making the measurements, right? And then validity we spoke about at length. We talk about the results. As we said, it can be presented in the form of an extract, a table, a graph, or a diagram. And now let's look at reliability again, improving the reliability. There's one that I didn't mention here that we should talk about as well. So we spoke about the investigation being repeated. We spoke about a bigger sample size being used. We spoke about the samples that are taken randomly. And no, so we didn't talk about that. The samples are taken randomly. That is another point that you can mention. And then many readings are taken to obtain an average reading. These depend on the nature of the investigation. Grade 12 is very important. You cannot just apply all of these to everything. Okay. It's like, for example, in planning steps. Okay, we're going to, my colleague Samantha is going to talk more about that as well. But like with planning steps, for example, you cannot just go and say, seek permission from the participants when the investigation is about plants. Right? Who are you going to seek permission from? From the plant. Right? So you cannot just say seek permission from the participant. You have to be specific in what you are saying. Okay? And it must be in context of the investigation. Then lastly, how do you come about the conclusion? It is directly linked to the aim of the investigation and confirms or refutes the hypothesis. So this one, you have to look at the aim and you basically answer the aim. So if the aim was talking about the influence of alcohol on the amount of urine produced, then you need to answer that question in your conclusion and say, what was the influence of the alcohol on the volume of urine produced? Right, now we go into the question. Okay, we've got a few questions to do. This one, as I said, what is the first thing we do? We read. Okay, we read and we already start identifying all of those things that I spoke about, right? All of those little circles that I was talking about. Right, so let's read through it together. And as we are reading, I'm going to start thinking about the aim and all of those things, right? Insects on islands without trees are likely to be wingless because flying is dangerous for them in an area with strong winds. A certain island was once covered with tall trees. Over the years, there has been deforestation in some areas of the island. On this island, insects called stoneflies have undergone natural selection. Right? That's a key word for me, is because they're telling us that natural selection took place. Right? And you know, when you think of natural selection, you should be thinking about who? Darwin. Right? Some have heavy wings and some have wings and some do not have wings, depending on the area where they are found. The diagram below shows stoneflies with and without wings. So now you can see what they look like. Scientists wanted to determine the relationship between the presence of tall trees and the wings on the stoneflies. So here we have an aim to determine the relationship between the presence of tall trees and the wings on the stoneflies. I'm going to write here the aim. And from the aim, we can be able to see what is the independent and dependent variable. So in this instance, what is the independent variable? It's the presence or the absence of the tall trees, right? So because that is the one that is going to determine what's going to happen, if there's going to be the wings of all the ones without wings, right? So the independent variable is the tall trees. I'm going to just put an I here. And then the dependent variable is the wings, the absence or the presence of the wings. That's the dependent variable. They selected 6 locations, of which 3 had tall trees and 3 were without trees. So they had 6 locations. And within these locations, they had 3. So that for me is what? Reliability. Yes, it is reliability. Because they didn't just take one location with trees and one location without trees. They took 3 of each category. Use a specialized net. So it was a specialized net that was used. They collected thousands of stoneflies. Right? Isn't that another one that you could say is reliability? Here we are referring to the sample size. So this is also reliability. What is a specialized net? They didn't just use any net. It was a specialized net. So isn't that got to do something with fairness? Right? Yes, it's got something to do with fairness. So this would be validity. The samples were labeled according to the area of collection. These samples were all collected in the morning during summer. So they were collected in the morning and they were collected during summer. Those are also constant variables in fairness, right? So you could say those are constant variables and it's all related to validity. Okay, we don't know what the questions are. We are just looking at this for now to see what am I understanding what is going on now. Because the fact that they were collected in the morning is important because they were all collected in the morning. It's not like it was any time of the day. They are being specific to say that they were collected in the morning, and also the fact that they were collected during summer. The number of stoneflies with wings and without wings at each location was counted and recorded. Okay. So now we've identified some constant variables, some validity, some reliability. Now we are able to answer the question. And now you will find that now that we've engaged and we've dissected this whole question, and we started thinking about what is the aim and the validity and reliability and the constant variables, then now when we answer the questions, you're going to see how easy it is. Identify the independent variable. What did we say it was? I hope you got it right. The presence or absence of tall trees. Okay, that is in brackets, basically the fact that they're tall trees, and the dependent variable are the wings on the stoneflies. The absence or the presence of the wings on the stoneflies. Okay, state 2 factors which were kept constant during the investigation. Now remember I told you about the constant ones. We spoke about it. There were quite a few. They use a specialized net, so the type of net, or you can say the apparatus used to catch the insect, the season of collection, or be more specific to say the collection was done in summer, the collection was done in the morning, or you can say the time of day of the collection. Remember, it's a variable. Okay. Type of location for each category, the number of locations for each category, the fact that it was 3 and 3. Right? Remember, there were 3 per category. Labeling of samples was done according to the area of collection. Okay, so that is all the constant variables that they were done. All of these things were kept constant variables, and all of these are going to ensure validity. They didn't ask us that, but this is just for teaching purposes, right? Say 2 ways in which the reliability of the investigation was ensured. How in which the reliability was ensured. Ensured is telling us what did they do. Where do you get it from? The information. Right. So what was it? They collected thousands of stoneflies. So which comment was that? I mean, which criteria was that? The generic statement is a large sample size was used, right? So collected thousands of stoneflies, and then 3 locations for each category. Spoke about that one. Right. Now we come to the 2 content-based questions, which I'm quickly going to do with you. And the reason why is because these questions were asked in a different way. It was like a new way of questioning. So I think it's important for us to go through it. Explain why it would be expected that more stoneflies with no wings will have no wings in the areas without trees. So now we're not looking at the investigation part. We're looking at the first part of information that was given to us. And they want to know more stoneflies that will have no wings in the area without trees. Okay, so now you've got to think about Darwin. Why am I saying that we are thinking about Darwin here? Because remember, they told us it's about natural selection occurred. So the first thing you need to ask yourself is, why would they have no wings without trees? What was the problem that they told us was the issue with areas without trees? Flying is dangerous in areas without trees. Remember, they told us that, right? However, the stoneflies with no wings survived because they could not fly. So what the whole thing is about is that where there are no trees, if they fly, it is very dangerous. So now they're saying that the ones that did not have the wings, they are the ones who survived because they could not fly and go to that dangerous area. So they then reproduced and they passed the allele for no wings to the next generation. The next question. Describe how Lamarck would have explained the evolution of stoneflies without wings in the areas without trees. Okay? So for this one, it is about Lamarck. So Lamarck is not Darwin. What did Lamarck say? 2 things. He said he had 2 laws. The law of use and disuse and the law of acquired characteristics. Okay, so now basically what does this mean? How is this different from what Darwin said? What's different is that Lamarck said that in this instance he's saying the evolution of stoneflies without wings. Okay, so he's saying that eventually they did not have wings. Right. So which wings they don't have wings. So in the beginning, they did have wings. That's what he's saying. So now they do not have wings, but in the beginning, they did have wings. That is what Lamarck is basically saying. And then because of the law of use and disuse, they use them more or they use them less, and then something else happened so that they could acquire that characteristic. Right. So what you need to ask yourself first with Lamarck is, what is the characteristic, the original characteristic? All stoneflies had wings. Then what happened? There was some sort of competition or selective force or something that happened that caused change. Due to the strong winds, they stopped using their wings because it was too dangerous to fly, right? They stopped using their wings. So their wings became smaller or they disappeared. And then that acquired characteristic was passed on to the offspring, the acquired characteristic. It's different from Darwin that he's talking about an allele. And all the offspring in the next generation now had no wings. So what I want to bring to your attention here is you see how they differ. They differ in that, firstly, Darwin had variation in the species and Lamarck had they were all the same. And in Darwin, the allele for no wings gets passed on to the next generation, whereas in Lamarck, it's the acquired characteristic that they get from not using it or using it too much, and that is what gets passed on to the offspring. Then I want to remind you that Lamarck's theory was the one that was rejected. Right, then we're quickly going to go on to this other investigation. And in this one, we're going to do a bit quicker because remember now we've already been doing the other one where we've been going through it slowly. Now we need to think again of the aim and everything, but a little bit quicker. An investigation was done to determine, as soon as I started to determine, I start underlining. This is the aim, right? To determine the relationship between the height of the head and bite force in lizards. What is the process? The procedure. The scientists collected 120 lizards with similar characteristics that were around the same reproductive age in different habitats. Their body characteristics and DNA were analyzed to determine if they belong to the same species. 40 lizards belong to species A, 36 to species B, and 44 to species C. So they were different species, right? Each species was kept in a cage with environmental conditions similar to their habitat. This is what the environmental condition is. The height of the head was measured for each lizard and averages calculated for each species. Using a Kistler force, the bite force of each lizard in each species was measured 5 times, and the average calculated for each lizard and each species. Okay, so here we have the results. We've got the species, the height of the head, and then the bite force. Let's look at the questions. Right, so they had, okay, before we do that, we identified the aim, like I said. Let's identify the independent and dependent variable. So the height of the head is going to be the independent variable, and the bite force is going to be the dependent variable. Right, independent variable, the height of the head, the dependent variable, the bite force, okay, because the bite force is determined on the height or dependent on the height of the head. State 2 factors that were kept constant for this investigation. So there were quite a few there that were kept constant. Yes, I underlined them. There were similar characteristics, same reproductive age, same measuring tool used for the bite force. Each species kept in the environmental conditions similar to their in habitat. I want to pause here. You would not have gotten the mark if you just said each same environmental condition, because it was not the same environmental condition. It was the same environmental condition similar to their habitat. Be specific. If they were specific in the investigation and they asked you what was kept constant, then you need to be specific. The last one, the lizards of the same species in each group. So all the lizards were not the same species, but they kept the same species together. So you cannot just say the same species. You wouldn't have gotten a mark. Okay. So Charles, I chose this one to show you that you need to be specific. Okay, and I'm explaining to you why. Apart from the sample size, state one way in which reliability of the results was ensured for this investigation. So remember, they told us there were 120 lizards. So they're telling us that a large sample size was used. Now they want to know, state one way, another way, apart from that one. So it's not your own thing that you need to make up. You need to look at the investigation. And what was it? There were 5 measurements of the bite force that were taken. The height of the head was different in each species of lizard. Name the type of variation displayed by this characteristic. The type of head was different in each species. It wasn't that it was 10 and 11. They weren't specific. Okay? But the type of variation here then is continuous variation, because they didn't have specific categories. There was a range. Right? Because there was a range in height. Describe the relationship between the height of the head of the lizards and the bite force. Now we need to describe a relationship. It's a description. What is the relationship? Lizards with an increased head height have a stronger bite force, or lizards with a decreased head height have a weaker bite force. Very important the words you used here. When we are talking about head height, you can use the words increased and decreased. But when you are using a force, when you are talking about a force, you cannot say an increased force or the decreased force in this instance, because it was about the bite force. The bite, as in when you bite yourself as well. It's a stronger bite force or a weaker bite force. So that was very important here. Also take note that when they ask you for relationships or they ask you for conclusions, it's going to be 2 marks, all or nothing. Oh, and then one other thing, when you're looking at this, you need to take into consideration that when you're describing the relationship, you need to look at the 2 variables. They're even telling you here what the variables are. But you need to look at them and you need to tell me what is the relationship between them. So the investigation was to determine what is the relationship between the 2 variables. Now, when they are asking you to describe this relationship. It's similar to when you're going to be asked to give a conclusion. Right. Are you ready for the next question? Right. The table below, this one is on genetics. The table below shows the results obtained in an investigation where a purebred black mouse was crossed with a brown mouse. The gene for black fur is dominant over the gene for brown fur. The F1 generation was then used as parents and 4 breeding pairs were used, and the results of the F2 generation can be seen in the table. Okay. So let's look at the parents. So we have a black mouse, and we have a brown mouse. And what we can notice over here is that in the first generation, all of them were black, right? And then we don't have any that are brown. Okay, so this should give us some information. And then what they did with the F2 generation, remember what they do with the F2 generation is that they take the F1 generation and then they interbreed them. Okay, so in this instance, what did we find is that in the first breeding pair, we had 8 black mice. In the second breeding pair, we had 5 black and 3 brown. In the third, we had 3 black and 3 brown. And in the fourth, we had 8 black and 2 brown. So from the data collected, calculate the simplified phenotypic ratio of black mice to brown mice in the F2 generation and show all your calculations. So they want to know a phenotypic ratio and it's of black mice to brown mice in the F2. So we go to the F2 generation. This is the F2 generation. So we have to add all of these up, right? So they want to know the ratio of this one and this one. Phenotypic meaning what they look like, right? So in our instance, this is the color. So we're going to take 8 plus, this is the 8 plus 5 plus 3 plus 8. Then we've got the ratio over there. The colon there to indicate to us it's a ratio. And then we're going to say a 0 plus 3 plus 3 plus 2. You get 1 mark for that. And it's divided, and this is now translated then into 24 to 8, right? That's the ratio, 24 to 8. And then you need to simplify it. So then 8 goes into 24 3 times, and that's how we get 3:1. So we have 24:8, which is simplified to 3:1. Now the next question is asking us, provide evidence from the data collected that supports the following statement. The gene for black fur is dominant over the gene for brown fur. Now, they told us this, right? But now they want evidence from the data collected. So how are we going to go about this? So when they ask you for things like this, it's similar when you're going to do a phylogenetic tree. The first thing that you need to look at is what are the parents? Then you need to look at what are the offspring. And why am I saying this? Because the offspring get, for each characteristic the offspring has, they received 1 allele from the father and 1 allele from the mother for that characteristic. I'll say that again. For every characteristic that the offspring has, they receive 1 allele from the father and 1 allele from the mother. So that can give us an indication of what the parents were. So in this instance, let's look at the offspring. Right? The offspring in the F1 generation, we've said that they were all black. Right? But now, they have 1 black mouse parent and 1 brown mouse parent. But all of them are black and none of them were actually brown. Right? So, this then indicates to us that if they had received the 1 allele from the mother and the 1 allele from the father, they both received the allele for black mice, which means that the one parent had to have black and the other parent, they had to receive at least 1 allele. I'll say that again. they had to receive at least one allele for the black color from one of the parents. So if this offspring is going to have black, let's say black is capital B, then the parent would have had black, right? The one parent. This parent could not, the brown mouse could not have had the black allele. Okay. Because, I mean, while it does not, you can see it doesn't have the black allele, and all of the offspring were black. Therefore, this one had to be heterozygous, and this parent had to be homozygous, and this parent had to be recessive in order to have the offspring being all black. Okay, and this is coming from our law of dominance. What is our law of complete dominance? We have our law of complete dominance. This is when one allele is dominant, it masks the recessive allele, and it further goes to say that if we have a homozygous dominant parent crossed with a homozygous recessive parent, all of the offspring will be heterozygous and for the dominant allele. Okay, so how do we explain this with answering the question? They just want us to provide evidence. They actually don't want us to explain anything. I'm explaining to you so that you understand how we got to the answer. And the answer is that the parents were black and brown mice, or you can say they were homozygous with contrasting characteristics, or you could have also said the one parent was homozygous for black and the other one was homozygous for brown, and therefore the offspring generation were all black mice. Okay, so that's how you explain it. Although that's what you state, but I was just giving you also the explanation behind it. Now, we come to a bar graph. Draw a bar graph to show the phenotypic results of the F2 generation for each breeding pair. You're going to get a graph in paper 1 or in paper 2. I can't tell you which graph, I can't tell you which paper it's going to be in, but you are going to get a graph, okay? Either a line graph, a bar graph, a histogram, or a pie chart. In this one, they are asking to draw a bar graph about the phenotypic results of the F2 generation. Let's just quickly go back to the F2 generation so we can see for each breeding pair. So you need to do it now for each breeding pair, right? So how are you going to go about this? Let's quickly just flag it. There are two ways to do this graph. I'm going to explain both of them. Okay, so firstly, let's talk about the caption. Okay, the caption then is this part over here. This is the caption. Right. How do you identify the caption? You need to tell us basically what the graph is illustrating. The number of black and brown mice for each breeding pair of the F2 generation. Grade 12s, you need to be specific. Your caption must contain all variables. Look here what it says. It says caption of the graph. It gives the marker an indication of what they need to look for. Does the caption have the number of mice? Does it have the color and the breeding pairs? Because the question asks you for the phenotypic results, which are the colors, of the F2 generation, right? So that is the breeding pairs, and then it's of each breeding pair, and obviously the number, right? Because you're talking about the number here. So it's very important that you have all of those characteristics. In some instance, they are going to have a relationship between the variables that will also be included in your caption of your bar graph. But in this question, there's no relationship. They are just wanting to know the number of black and brown mice for each breeding pair of the F2 generation. So you can do it one or two ways. In this one, what they chose to do here is the number of mice. You can see in both instances, the dependent variable is the number of mice. But in this first one that we're looking at on this side over here, this one, they decided to do it, have a key for the color of mice. So the independent variable is the breeding pairs, and then the black and the brown mice are the different colors. And then that's how they chose to show it. So they have the first breeding pair, there were 8 black mice and there were 0 brown. The second breeding pair, 5 and 3, and it carries on like that. Whereas this graph, they chose to do the breeding pairs in different colors, or the keys for the breeding pairs, and the color of the mouse is on the x-axis, which is the independent variable in this instance, and then they have chosen to do it in this way. Now, what I want to bring to your attention is, when you are doing a bar graph, it is important that the space between the bars here are equal. These two bars can be right next to each other. It doesn't mean that this is a histogram now, because these two bars are next to each other. But for each category represented here, the space between the pair of bars must be equal, and every bar must be equal, right? The size. So how are you going to do this if you don't bring a ruler? So many grade 12 learners don't bring a ruler to the exam. A ruler with numbers, a ruler with markings on it. They don't do that, and this is why they lose marks. How are you going to measure 2 centimeters with the naked eye? Okay, so this is what you need to do with the histogram, and the bar graph. Your bars must be the same width, and the spaces between the bars must be the same. When it comes to this one, you see what's happening here. Everything for this category, they are all together, but there's a space between these ones. Very important. Oh, sorry, not even till there. Sorry, it goes till there. There is a space between that. Okay, this one is a 0. And also, can you indicate here that the 0 is here? The 0 is indicated here. Yes, they gave you 0. You need to indicate 0 there, grade 12, because it is a value. 0 is a value. Okay. So, like I said, the bar, you're going to get a mark for T for the bar graph, for the type, the caption, you're going to get a mark for a caption if you have all the variables. You're going to get a mark for labeling the X and the Y axis. In this instance, there are no units, but sometimes there are units. You please need to take care to look at that. The correct scale. What do they mean by that? They want us to have the correct scale for the y-axis and equal spaces between the bar and equal width of the bars. That's what I spoke about on scale. And then the plotting. How many bars did you plot and if you plotted them correctly? Right, so we're going to go to a quick break, and please stay tuned. We're going to have a very quick break, just 2 minutes, and then my colleague Samantha Raghunandan is going to come back and complete this lesson on scientific investigations. That's all from me. Thank you.
afternoon, grade 12. Welcome back to the second half of our lesson on scientific investigation. My colleague Vedika has taken you through the basic concepts of this topic, and I'm sure you've realized by now that they can place a scientific investigation on literally any topic in your syllabus. So you need to know the scientific skills, and also you need to know the scientific content. We always talk about learning the content is not simply enough. In every single life science paper, there must be a scientific investigation. So this is a skill you need to master. So we're going to continue with example, but I'm sure by now you have realized that the basics of every scientific investigation, they have common exam questions. So these are things you can prepare for. Every year, they would ask basic concepts in scientific investigations, be it what is the aim of the experiment? What makes the experiment more reliable? What makes it more valid? What can you conclude from the experiments? And almost always there's a graph or there's a response to a graph. So if you have not brushed up on your graphical skills, grade 12, you need to know how to draw a graph. So please, that is very, very important. We have covered some of it, but obviously because of time constraints, we can't cover all of it. Okay? So if you must know how to draw a pie chart, you must know how to draw a histogram, you must know how to draw a line graph, a bar graph. And there's a section in your mind the gap that addresses this. So if you don't know how to do all of this, so please, after this lesson, you need to do this. So while I'm doing the question from the June exam 2022, I want you to have a pen and paper in your hand because you're not going to be able to remember certain concepts, and you need to remind yourself as you are studying. Let's read the experiment. Okay, it says the human reaction times can be tested using the apparatus in the diagram below. So what are they testing? They're testing the reaction times using the apparatus in the diagram below. There we have a picture of a laptop. It's plugged into a light, and we also have a button that they are pressing. So obviously we're going to look at the diagram, and the diagram is going to give us information about the concept. So when the investigator starts the computer program, the light flashes randomly. The person being tested, the person, so already we can see that there is one person being tested, presses the button with each flash. There we go. So the time taken for the reaction, that means the time between the light being switched on and the button being pressed, is recorded in milliseconds. So they're giving us a lot of information. Excuse me. So when it comes to a scientific investigation, you always must read the stem of the question, as my colleague has spoke about. You must read it because some of your answers is coming from the question. So it's important that you read it and you take the text. So before we can even go to the questions, because sometimes when we look at the questions, it can confuse us a little bit. So let's look at what is basically going on here. If we look at it, we got the light. They switch on the laptop, okay? And every time they start the computer program, they measure the time that the person, so the light is switched on first and then the person presses the button. So they're measuring the time that it takes for the person to respond. And what is the unit that it's measured in? Is in milliseconds. So you must take note of that because that is going to form part of your answer. So we know more or less what they're going to ask, but let's see what are the questions. So state the aim of the investigation. Now, based on our content knowledge, we know that the aim is usually given in the beginning of a scientific investigation, and most of the time it includes both variables, the independent variable and the dependent variable. So with regard to the independent variable, how do we remember this? Because we must know this before we even start asking the questions. We must know that the independent variable is the variable which is controlled by the investigator. So whatever the person who's running the experiment, whatever variable they are controlling or what factor they're controlling, that is going to be our independent variable. And from our knowledge, we know that the dependent variable is the one, is the factor which reacts or responds to the independent variable. So let's look at the aim. With that background knowledge, we're going to go forward now, okay? So the aim is to investigate the time taken for the reaction to take place in milliseconds. You can already see there it's got two variables. It's got time and it's got the reaction. It wouldn't be correct enough if we said to investigate the time taken for the reaction to take place. In what? The time could be days, it could be months, it could be hours. So we need to be specific. We're saying in milliseconds. And you'll notice that there's two ticks at the end of the sentence, grade 12. What does this mean? It means that this is 2 marks. Besides that, it also means that the entire sentence must be written in order for you to obtain 2 marks. If you don't write that answer as it is, if you don't include the two variables, if you're not specific about the milliseconds, you won't get no mark. Okay? So please, you need to pay attention to detail. And as my colleague said, you need to be really specific. Okay? Let's identify the independent variable. Remember we said, what did we say the independent variable was? We said it is the one that is controlled by the person controlling the experiment. And obviously, this would be the time. Because remember, the person who is starting the computer is controlling the time in which it takes for the person to respond, the second person to respond. So the dependent variable would obviously be the reaction. So the dependent variable would be the reaction. And most likely we would find the dependent variable on the y-axis because this is the value that is going to be changing. It's going to be responding to what is happening in the independent variable, which would occupy, as we know, the x-axis. Give three planning steps that we should be considered when conducting the investigation. Now, grade 12, this you do not have to just take it out of your mind. The question gives you some of the answers. You need to look at what is actually going on in the experiment. And you need to plan, because we don't just walk into the room and you say, okay, we're going to do this experiment. There's preparation involved. Let's see what type of preparation is involved. We need to decide on the sample size. Why do we need to decide on the sample size? We need to decide on the sample size because we cannot have a sample that is too small or a sample that is too large because it would take longer. A sample that is too small would not be reliable, okay? Obtain informed consent or permission from participants. Obviously, if there's human beings involved, you need to get their permission so you don't get sued, okay? So you have to get the person's permission to run this experiment. Decide of the place to conduct the investigation. Now, this is important. We need to take into account where this experiment is taking place. Remember, this experiment is measuring reaction time. So can we conduct the experiment in a place that's noisy, that there's distractions going on, that there's people walking around all the time? The reaction time is going to be affected because the person who is undergoing that experiment, the participant in that experiment, is going to be distracted. It's going to affect his reaction time to pressing that button. So do you see how important the planning steps are? It is vital, and you must know this. You can't just say, oh, I wasn't there, I don't know how they planned it. The experiment, the stem of the question, will give you an idea of what is needed to be planned. The duration of the experiment. We can't have an experiment that is running too long, because what happens if the experiment is too long and the person becomes fatigued or tired? Their response time to pressing that button is going to be slower. It's going to affect the experiment. So the duration must be reasonable. The person to administer the investigation. And this is very, very important because there needs to be the same person who is running the experiment. Why? Because of human error. I come and do this experiment. Maybe I press a few buttons before I start the computer program. The person B comes to conduct the experiment. Maybe they do something else before. If the same person conducts the experiment, they will conduct the experiment in the same appropriate manner. But if we have different people, there is going to be human error involved, and our experiment is not going to be valid. Okay? A person and the age of the person, obviously, an 8-year-old is going to respond differently to an 80-year-old. So this must be taken into account. The gender, whether it's male or females that you are going to be deciding on. Decide on how to measure the results of the apparatus to be used. You need to decide, as a person conducting the experiment, how are you going to show your results? Do you want to show it in the form of a table, or do you want to show it in a different form? Recording tools, methods of representing the data. Do you want to maybe convert this table into a graph to display your results? So those are all planning steps. Do you see, great job? This is so simple. This is common sense, common knowledge. This is a section that you don't need to be scared of. It really is so simple. We've got the guidelines. We need to know the aim. We need to know what is a dependent variable, what is an independent variable. I mean, these things are concepts that we just have to learn the definition and we apply it in every single situation, because you can see you can get it under any topic and they'll throw some content in it just to shake it up a bit. Okay? So, next one we're going to go to is the Gauteng Prelim 2024. So from all the examples that we've done so far, great job, you'll notice there's no set way in which they give this experiment. Do you see the one that I had before was a diagram? It had pictures and a little bit of a write-up. And then now I'm getting a paragraph to read. But I like paragraphs. You must love paragraphs because then you're getting more information. They're giving you more answers in which you can work on. So let's read what they say. A student studied the effects of various plant hormones. Do you see? You can put it into any section. Plant hormones on the growth of corn seedlings. Corn seedlings of the same height. This is important because we know that in order to make the experiment fair, we have to have the same sort of the same sort of procedure. The student took 4 groups of 10 plants each. All the initial heights were 1 centimeter. That's important because if I've got plant hormones, obviously already I know at the back of my mind, this experiment is going to be on how they're growing. Which one is growing faster, which one is growing slower. So if I start with a corn plant that is 2 centimeters and 5 centimeters and 1 centimeter, how am I going to measure? Okay, it's not going to be reliable. Okay, each group received the same amount of water. They must receive the same amount of water. Why? If I give more water to one plant, will it not grow faster? So these are all taken. Can you see all of this is a procedure? This is all the planning steps. This is the scientific procedures which make the experiment valid. Okay? She gave one group water only, while the other groups each received 1 microgram of different hormones mixed with the water. After 15 days, notice there's a time frame. After 15 days, she measured the height of each seedling from each group. Calculate the average recorded information in a table below. Excuse me. This is going to be so simple because already you can see there's validity here in this experiment because the procedure that is being followed is validity is improved at grade 12 when we eliminate those factors which can influence your experiment. And only the variable that is under investigation is changed. So they are trying as far as possible for this experiment to be fair and valid. Sorry. So let us look at the Gauteng Prelim 2021. They give you a table there. I already can see this is a lovely, lovely question because they're giving you information. They're giving you a table. They're giving you on the one side of the table, they're giving you the various plant hormones mixed with water. So we already know one is water only, one is abscisic acid, and so on. But if we look on the other side of the experiment, other side of the table, we got the height, and we can see all these values are differing. Okay? Excuse me, great job. So all of these values are differing. So if we look at the various plant hormones, we can see that the factor that the investigator is controlling, would it not be this column, the plant hormones mixed with water? Because that is going to be our independent variable. The definition of independent variable, it is that factor which the investigator is controlling. And then the height of the seedlings. The height of the seedlings depends on what is being the factor which the investigator is controlling. So already we can see that this is going to be our independent variable. Okay? So let us look at this question carefully. Did you notice I did not even look at the question? I'm looking at the information given to me. based on the facts that I know. So let's look at the question now. Which plant hormone produces the tallest plants? How simple is this? We need to go, height is synonymous with the length and the height. So, which is the tallest plant? Let's number it. So, the greatest height here is gibberellins. The second one is going to be oxons, correct? The third highest is going to be water only, and the fourth is going to be abscisic acid. So now we already know which of these hormones that they treated these plants with was most effective in height. So now let's visit the question. Do you see how it all comes together in your mind as you just extract from what they give you? So if they're trying to trick us, they're not going to achieve it. Which plant hormone produces the tallest plant? Remember, we already discussed this, so we already know the answer. It's going to be gibberellins because it has 48 centimeters in height. Okay? Then the next question. Identify two ways that the student ensured the validity of the investigation. How did the student ensure that this is going to be a fair investigation? So here we need to look at what the procedure of the scientific procedure. Remember, the validity is measured by is improved by the scientific procedure that is followed. The same height, we have already discussed this, so I'm going to go through it. The height of the corn seedling. Remember, we said we couldn't do 5 centimeters, 1 centimeter, because it wouldn't be fair. Our results at the end would be questionable. Okay? The same species we use. Obviously, you need to use the same species. Why? To make it fair or to make it balanced. Because some species grow faster than others. Some species respond more maybe to the oxons than to the gibberellins. We don't know. So we need to make sure that when we start, the same species are used of the same length. The same number of seedlings in each sample. What are seedlings? We know seedlings are cuttings which are taken from not other stems but from seeds. Okay, they are seeds that grow and they grow into young plants. The same amount of water was given. The same. Notice same. You already know the trend. What's trending like TikTok? What's trending in validity? It must be, most of the things must have the same sort of structure. So the amount of hormone in the mixture, the same period of 15 days. Because think about it, grade 12. I leave this experiment to go on for water only. I leave it to go on for 30 days. For gibberellins and water, I leave it only for 15 days. It's going to affect my results. So the time frame in which we are working with needs to be, yes, you said it, the same. Do you see how you can find tricks to remember all of this work? Right. What are the functions of abscisic acid? So there's two functions of abscisic acid, okay? Well, actually, there's several functions, but you're only going to get marks for two. Let's go through this. Right. Two functions of abscisic acid. Now, I like this question because very often in life sciences, a lot of us are not first language. English is not our first language. We are second language learners, and it becomes challenging to understand. So when I go through this, I'm going to explain what each term means, like stimulus, inhibits. What does it mean? So when you are answering in the exam, you will have an idea. For those of you who don't know, okay? So it promotes the dormancy. Dormancy basically means when the physical functions of it is slowed down. It promotes dormancy of seeds and inhibits germination. What is inhibit? Inhibit means it prevents. Okay? You can't just go write inhibits and you don't know what it means. Okay? It inhibits, it slows down the growth of apical bud. This is all biological terminology which we use. But you need to understand. It induces flowering in plants. What does induce mean? It brings about flowering in plants. That means it causes plants to flower. It promotes aging of leaves. It stimulates the abscission of leaves. That means it causes leaves to fall off. It causes the closing of the stomata. So any two of those is going to be correct. Okay, I'm not going to spend too much time because we are focusing more on the scientific investigation and how we can interpret the graph and how we can understand what is the variables and how we can get information from a table. So if we move on, draw a bar graph to represent the data of the table. So grade 12, do you see I was not lying to you? When it comes to scientific investigation, graphs, tables, diagrams, they're very, very important. So you have to know how to draw the graph. So what I'm going to do is I'm going to give you the graph and the criteria. My colleague has already alluded to the fact that the spaces, you can't go and draw one thin bar graph and one fat bar graph. You need to be standard. Remember, we are scientists. Everything must be, it must be meticulous. It must be according to the data you are received, and it must be according to scale. So, what are you getting a mark for? Now, I did this example because I want you to understand when you are writing the caption of a graph, it doesn't mean you must just take the x-axis because that's the independent variable, and you must just take the y-axis because the x-axis is the independent variable and the y-axis is the dependent variable. We can't just take those 2 points and put them together. Your caption needs to make sense. Let's look at the caption of this graph. It says, bar graph showing the effect of various plant hormones on the growth of corn seedlings. So there's the various plant hormones, there's the growth of corn seedlings. But notice, as my colleague has said, we need to be specific. Imagine if we had to write the heading, bar graph showing various plant hormones on growth seedlings. So what? Effect is something, it is when an action is taken and it brings about change. So in your caption, it must be a description, a detailed description of your x-axis and the y-axis, and yes, the relationship between the two. Okay? So if you did not write that in full, you would not get a full mark for the caption of the graph. You are getting marks for the correct type of graph. You can't just go now draw a line graph because that's a graph you know. You're going to get no marks for that, for the type of graph. Okay? And then you're going to get marks for the axis labels. So it must be in full, and you can see for our dependent variable, usually there's a unit, and that unit must be included because we need to know what units we are measuring in. And then you're going to get a mark for scale. You can see there's even spaces between the bar graphs. There needs to be, because if they're all together, then we are dealing with a histogram, and we don't want that because they asked us to draw a bar graph. Okay? And then the plotting of the points, I think that's self-explanatory. You will take the data from your table and project it onto your bar graph. It needs to be neat. You need to I'm saying it again, my colleague has said it, but you need to bring the necessary instruments. You need to go and bring a projector, because what happens if you get a pie chart? You need to bring a ruler, you need to have a pencil and an eraser. What happens if you make a mistake? Okay, grade 12, basic things. I know you were stressed out, but you need to remember all of this, write it down so you don't forget. Okay? So let's look at the next one. Do you see how you can get it in different forms? First, we got a diagram, and then they gave us a table and little extract. Now they're starting off with a graph. So these are different ways in which they can ask you. But we're not worried because we already know our basics. We know how to identify our independent variable. We know how to identify our dependent variable, and we're good to go. Okay? Let's start. An experiment was set up to investigate the diameter of the pupil. There we're going to respond to the environment by humans under different light conditions. So the question is already giving me information. An experiment was set up to investigate the diameter. So what are they investigating? The size of the diameter of the pupil under different light conditions. A person sat in a dark room, that's key, and an electric bulb was switched on at different distances from the person. So basically, if the person was sitting there, they switched on the light here, then sometimes they switched on the light here, in different positions. So the person's eyes needed to adjust. Remember, this is not something that is abstract. Life sciences is such a wonderful subject because we can relate to it. It's everyday life. Okay? So you can picture it in your mind. And then The diameter of the pupil was measured how long? 1 minute after each time the light bulb was switched on. So this is reaction time, yes, but reaction time of the dilation of the pupil and how wide the pupil has been opened. The results are illustrated in the graph above. So let's see. Here we know that the position of the light source is on the x-axis. So what is this position of the light source? It's explaining the different light conditions, isn't it? We've labeled it position of the light source, but we know what is this depicting. It's depicting what they said in the question, different light conditions. I will unpack that just now, but I want you to take note of that. And then if you look at the diameter of the pupil, it varies. So at some, we can already see here, at some points, the pupil's diameter was very low. It was only 2 millimeters. So that means that the pupil did not open as wide. That means the light was closer. But remember, we're dealing with a dark room. So if they put the light somewhere far away, then the diameter is going to open a little wider to adapt to bright light. To light in a dim area. Okay. So let's look at the questions. Name the following for this investigation. I put the graph there so we can reference it. So you must always go back and forth when you are answering the questions and look again and double check. Okay. So name the following for this investigation. The independent variable. Okay. So from our knowledge, the independent variable, what is it? It is the one that is controlled by the investigator, the person who is running the experiment. I'm saying it over and over again, not because I'm old, grade 12, because it is something we have to constantly say to ourselves before we start. Okay? We are reminding ourselves. So we said that it's controlled by the investigator. So let's think, what was controlled by the investigator? The light, isn't it? The light conditions. It was all over the place, like the diagram I drew. If we say position of the light source, we won't be correct. What happens if that position was the same all the time? What happens if we put the light, the person sat there, and we just put the light there? So it wouldn't be correct. Do you see? It's not just a random answer you can give on a definition. You need to understand the context of the question. So the independent variable in this case would be the different light conditions, because that is what the person running the investigation is controlling. The dependent variable we know is on the y-axis, and that would be the diameter of the pupil. So please, grade 12, don't just assume. You need to go back. You need to read the stem of the question. You need to see exactly, hey, what is this investigator controlling? What is he not controlling? Okay? State 2 reasons why the results of this investigation is not reliable. Notice how they try to trick us here. Usually they say, what makes an investigation reliable? So now we just need to turn it around. So what is our first thing we do? Before we even think of the answer, okay, what makes the investigation reliable? What makes it reliable is the fact that I always remember reliable, repeat. Reliable, repeat. That means the investigation is being repeated. Okay? Also, that there is a large sample size taken. That's what makes it reliable. Okay? So what's going to make it not reliable? Remember, was the experiment repeated? No, it wasn't. If we go back to the stem of the question, they said a person was sitting in a room and then they moved light around. They didn't say a whole group of people or 15 people or 20 people, a person. So that means one person was used to conduct this experiment, not a very reliable experiment, in my opinion. Okay? So you already know the answer to this. Only one person participated in the experiment. So as much as you know what is the definition of reliable, you must also remember what makes it not to be reliable. And they can also do this with validity. So you must be able to think on your feet, grade 12. You understand the concept. Just apply. Okay? So one person was participated in the experiment, and also the experiment was not repeated. I hope they give you this question because it's such an easy question, easy mark. Because you know what is reliable. All you have to do is tell them what was not reliable about it. Okay? Right. Calculate the percentage increase in the pupil diameter when the light bulb moved from position 3. Now you must physically go to the question paper. We're talking about point 3. They say calculate the percentage decrease. Usually they give us percentage increase. So this is important because you must read the question. You must circle, you must underline, you must make notes on your question paper so that you can understand and make sense of it. When the light bulb was moved from position 3 to 4. So from here, the light bulb was moved to 4. Can you see what happened? Let's look at it before we even look at the calculation. Was it increased or decreased? It was decreased because you can see the graph is going down. Okay? And we know when we are calculating percentage, we know that we must multiply whatever we're doing by 100 to convert it to a percentage. We know that. Okay? And we know that because it's percentage decrease, we're going to subtract the one number from the other number. Okay? So wait, from position 3 to position 4. So let us look. So it's going to be, we're going to look at the value. So what is the value? Again, here again. Am I supposed to be using? You should be shouting at me. You're supposed to be saying, ma'am, you're supposed to have a ruler. But because for the interest of my learners in grade 12, I'm going to make a dotted line. So let's look. That is position 3. Position 3 is 8 millimeters. So there's position 3. Okay? And position 4 is 5 millimeters. There's position 4. So we subtract. So we calculate the difference between the two diameters, the size of the diameters, and we express it over the initial diameter. Okay? And then we get our answer of 37.5%. So that is how you calculate percentage increase. So do you see? They can throw a whole lot of things. They can even ask you to calculate. They can ask your calculations. They can ask you graphs and all of those things. Okay. So let's look at the next question. Which colored structure of the eye is responsible for changing the diameter of the pupil? Now here we go into content. We need to know the structure of the eye. And we all know that the answer to this is the iris, isn't it? Because the iris is that part of your eye that has the eye color, and it also has, besides the eye color, that is unique to you. No one else has got that eye color. Do you know that? It's actually unique to you. And besides the fact that it's so beautiful and it's got that eye color, it has muscles. And these muscles make the pupil smaller and bigger. Okay? So that would be the iris. Name the mechanism. Now, keywords, mechanism. So there's something we know which mechanisms we did. So already we know the answer, but we're not going to talk the answer now. Because when we did the eye, we did two processes that are involved. And one of them was a mechanism that caused the eye to change in the diameter of the pupil from position 2 to position 3. So here we need to go back. I'm just going to go back. Yes, there we go. Yeah, so it changed from position to 2. So it was first here and it changed to position 3. So what happened there? Can you see the pupil was much smaller? So that would mean the light was closer. Remember, we were working in a very dark room here from the question. And then all of a sudden the pupil became bigger. And we know from our work under this certain mechanism, we know that when it gets dimmer the light, then the pupil needs to adjust. But what is this secret mechanism we are talking about? It is the pupil mechanism or the pupillary mechanism. That is, pupillary mechanism is what we are used to. So that is an easy answer, marks for nothing. You just have to name the process. Okay? Describe the mechanism that is mentioned in question 5. So remember we said that the pupils are going to get much larger. So we need to discuss the process. The circular muscles of the iris relax. The circular muscles. Circular. And we know which is the radial muscles. Isn't from our maths work. Radius is from the center to the circumference. So that is our radial muscle. So the radial muscles of the iris, they contract. And the pupil diameter increases because it's adjusting to the light. Okay? And if we move on, give the diameter of the pupil at the position 4 of the light source. So there's position 4. Great jobs. This is so simple. It's just reading of graphs. So I don't understand why you were so scared of scientific investigation. Because do you see now? They give you all the answers. You just need to remember your concept. Okay? So diameter of the pupil at position 4. Now, what are we going to do? We're going to use our ruler and we're going to check what is the diameter. Yes, position 4. We're going to go to the side. You see, because I'm not using a ruler. But what is that going to be? It's going to be 5. Yes? No. Why not? Because it needs to be 5 millimeters. Remember, whenever you are reading off a graph and the graph is values, you must always put, yes, the mark is on 5. But in some cases, they might give a mark, 2 marks for the millimeters as well. So 5 millimeters. And the position where the light source was furthest from the person. So where was the light source furthest from the person? And that would be position 3. We know here because the size of the pupil became much larger to adapt to the dim light, isn't it? So that is all for today, grade 12. But I'm hoping that you now realize what a lovely section this is. And the good thing is that you can get marks here, in some cases, for not even, sometimes if they give you a scientific investigation on a question that you're not so familiar with. But because you know what is an independent variable, because you know what is a dependent variable, because you are able to identify an aim, you are able to name a graph, quoting both variables, you're going to win at this. Okay? So, so thank you so much for tuning in. You can go through this lesson all over again on your own on the platform that you are watching it, and you can rewatch it in case you have some gray areas. But good luck and all the best for the final exams. I know you're going to do well. Thank you.